Hey everybody, we are going to continue our talk on photosynthesis, and we are going to first look at something called photosystems. So photosystems are collections of pigments that serve to trap light. And so we talked a little bit about pigments last time, and we talked specifically about chlorophyll, and then I also mentioned some accessory pigments, and so it's these chlorophyll pigments and accessory pigments that make up the photosystems. There are two types that we know about, so photosystem 1 and photosystem 2. You see here abbreviated as PS1, PS2. And this is just sort of a quick diagram of a photosystem. And so you can see we've got these pigment molecules. The pigment molecules serve to sort of harvest this light energy in the form of photons. That energy gets transferred from one pigment molecule to the next. And then finally it will hit the reaction center, which is going to be a chlorophyll molecule or chlorophyll pigment. And then you'll have electron transfer. So then the electrons from the chlorophyll will get transferred to the primary electron acceptor. So this is an example of a photosystem. This is what it would actually look like embedded in the thylakoid membrane. So here we have the thylakoid membrane. So this would be the outside of the thylakoid, so the stroma, and this is the thylakoid space. So you have light coming in. Again, it's going to cause an energy transfer from one pigment molecule to the next, all the way till it gets to the chlorophyll A. Then you're going to have the electrons move to the primary electron acceptor. And all of this is happening within this reaction center complex. And this is the larger picture then would be the light harvesting complex. And so this is happening within the thylakoid membrane in the chloroplast. All right, so the first thing we want to talk about is cyclic photophosphorylation. So this phosphorylation part, you should have kind of be familiar with that. So phosphorylation is anytime you're adding a phosphate group, photo meaning light and cyclic meaning it happens in a cycle. And so cyclic photophosphorylation is going to use only photosystem 1, and it does produce ATP. It also does require light. So you have your photosystem 1 right here. I've shown you this sort of in this larger complex. So here you have photosystem 2, but it's sort of grayed out because we're not looking at this. We're only looking at cyclic photophosphorylation, which happens with photosystem 1. You can see the light coming in here. You're going to end up sending 2 electrons up to this primary acceptor. The electrons are going to go through, this should sound familiar, an electron transport chain, so they will get transported from one protein to the next. They will make it to what's called the cytochrome complex. And here you end up with energy for the chemiosmotic synthesis of ATP. So you're producing some ATP. You are also sort of, like I said, it's a cycle, so then you're also going to have energy move back to photosystem 1 along with more light. You're going to have the electrons sent to the primary electron acceptor again. So this will go in a cycle, and it is a way to produce ATP. Next, we're going to look at linear or non-cyclic photophosphorylation. This is going to use both photosystems 1 and photosystem 2. It's going to split a water, and when that happens, it releases hydrogen, a pair of electrons, and oxygen. And this whole cycle is going to produce both ATP and NADPH. So the NADPH is another electron carrier. When it's got the 2 forms, so when it's not carrying the electrons, it is NADP+. When it's picked up the electrons, it will be NADPH. So it looks very similar to the NADH that we talked about in cellular respiration, does the same job. An easy way to remember the difference between the two is that NADP+ and NADPH have the P for photosynthesis, whereas then you have NAD+ and NADH. There is no P in that, so it is cellular respiration. Just a quick trick to kind of make sure you can identify which one works with cellular respiration and which one works with photosynthesis. So here we're going to start down here. I've got these numbered steps. So the first thing that's going to happen, you can see we've got that yellow line again. So you've got light coming in. Again, you're going to have some energy transfer until you get to the chlorophyll A, at which point step 1 then is, because of the energy from the light, 2 electrons are going to get sent to the primary acceptor. So that is step 1 in non-cyclic photophosphorylation. Step 2 is that down here you've got this water coming in. the water is going to be split. So you end up releasing oxygen gas and hydrogen. The electrons are going to go and replace the 2 electrons that were just sent up to the primary acceptor. So step 1 is the energy from light is harvested, and then 2 electrons get sent to the primary acceptor. Step 2 is that water is split and 2 electrons are given back to that chlorophyll to replace the ones that were just sent to the primary acceptor. Then you've got energy moving through this electron transport chain, so this is step 3. Step 4, here you are making some ATP, so you've got energy for the chemiosmotic synthesis of ATP, so ATP production is happening here. Then you're going to continue down this electron transport chain. Step 5, we're back at photosystem 1, so again, more light energy coming in, more excited electrons going to that electron acceptor. And then step 6, we have this electron transport chain again, and now here we are going to end up producing NADPH. So we have NADP plus reductase, so this is an enzyme that is going to convert the 2 NADP pluses and 2 hydrogens with 2 electrons to make NADPH. All right, so this is non-cyclic photophosphorylation or linear photophosphorylation. A couple of things I want you to pay attention to. You are making ATP. You are also making NADPH. The lines here are corresponding to the energy. So here you've got the light energy coming in, and so you've got like this increase in energy as the electrons go to the primary acceptor. They're spending energy as they move through the electron transport chain. They get re-energized by more light here in photosystem 2, so energy goes back up, and then again as you move through this electron transport chain, energy will go back down. So that's the reason it's kind of in that zigzag pattern. All right, so we talked a little bit about how we've got these light-dependent and light-independent reactions in photosynthesis. So the light-dependent reaction is the first one we're going to look at, and this is the same thing as non-cyclic photophosphorylation. So this is going to happen in the grana of the chloroplast, and the whole reason that we have this light-dependent reaction is to split water in order to produce ATP and NADPH. So this is happening in the chloroplast, you're splitting water, you're energizing these electrons in order to produce ATP and NADPH, which will be used later on in the light-independent reactions. So what do you need for the light-dependent reactions? You need light, first of all, because they're called the light-dependent reactions. You need the water that is going to be split. You are going to need some ADP and inorganic phosphate because you're making ATP, and so in order to make ATP you need to have the building blocks, and then you also need to have an NADP plus because you will be making the NADPH at the end of this. So please notice that you've got your requirements here on the left and they have corresponding products on the right. So water is split and that produces oxygen gas. ADP and inorganic phosphate are required because you're making ATP. NADP plus is required because you're making NADPH. So they are aligned. There is not a product correlated to light. Okay, so we want to talk really quickly about this chemiosmosis model. And so this says that the chloroplast produces ATP in the same manner as the mitochondria produces ATP in cellular respiration. And so now you're using light energy to pump hydrogen ions across a membrane. And then again, when those hydrogen ions diffuse back, ATP is going to be generated. So you're going to see ATP synthase pop up again. And it does look very, very similar to what we talked about in the last chapter. So you're going to have hydrogen ions pumped into the thylakoid space. With cellular respiration, we were looking at the hydrogen ions being pumped into that intermembrane space, so they were building up that hydrogen ion concentration quickly. Here this is happening in the thylakoid space. And then ATP and NADPH are made when the hydrogen ions diffuse into the stroma. So here's just a quick breakdown. You've got photosystem 2 and then photosystem 1. So this is that non-cyclic photophosphorylation that we just talked about, but now it's showing you how it takes place within this thylakoid membrane. So this is the membrane, this is the inner thylakoid space, and then this is the stroma. So this is just sort of the outside outside of that thylakoid, but still inside of the chloroplast. So again, light energy comes in. water is going to be split, so you're producing some hydrogen ions right here. You have the electron transport chain. You're passing these electrons down. As this happens, you are providing energy to pump some more hydrogen ions into that thylakoid space. Electron transport chain will continue. You will get re-energized at photosystem 1, more electron transport chain, and now you're making, you're at the NADP reductase, so you are going to make your NADPH right here, and that will get sent out to be used during the Calvin cycle. Now, if we look at the inner thylakoid space, what's been happening is that we've had these hydrogen ions pumped in and released from water, and so the concentration of hydrogen ions is going to grow in this tight little cramped space. In order to kind of get away from each other, these hydrogen ions are going to want to diffuse back into the stroma. The only way they can do that is to move through ATP synthase. As they move through ATP synthase, you will make ATP, and then again that ATP is going to be used later on at the Calvin cycle, which is the next part of photosynthesis. So just a quick comment, there can be a pretty drastic pH difference, like a 3 pH unit difference between that thylakoid space and the stroma, and again, you're looking at these hydrogen ions being pumped across the membrane to cause that pH difference. Alright, so your lecture question for today, shoot me some responses. I want you guys to compare and contrast how ATP is made in chloroplasts and mitochondria, so you guys could talk about the process that leads up to ATP production, talk about where it happens, talk about when it happens, but I want you guys to compare ATP synthesis in chloroplasts and mitochondria. So send me a message with your answers. Thanks, guys.